in the show a scientific enterprise, the first scientists to work there, and to accompany this company in the development of Anton from the very first, using it to study GPCR receptors, and now he's heading his own group at Stanford, and we'll hear about the latest work.
Okay. So first of all, thank you for the invitation. This has been a wonderful meeting so far. Hopefully I will keep it up. I got a couple questions just now about what the photo was. So this is a photograph of a 3D printout of a computer-generated model. Okay, and I'll talk later on about how we arrived at this model. But basically, this is a GPCR with two ligands bound to it, an orthosteric ligand and an allosteric ligand. So I'm going to talk today about molecular dynamics simulations. I'm not going to focus on methodology, in particular because several previous speakers, Michael and Tamara, have talked about methodology of MD simulations. I'm going to focus on applications to one particular biological system of interest, in particular G-protein coupled receptors, which represent the largest class of drug targets. They represent the targets of about a third of the drugs on the market. That is, about a third of the drugs on the market act by binding to a GPCR and affecting its function, usually making it signal more or signal less. So let me start with sort of a one-slide summary of how GPCRs work. The basic idea is that these are membrane proteins, and their basic function is to transmit signals from the outside of the cell to the inside of the cell. In particular, they sense the presence of various molecules like hormones or neurotransmitters on the outside of a cell, and they send that information into the cell and stimulate intracellular signaling in response to those molecules. So the way this works is as follows. Those molecules, for example, hormones and neurotransmitters, bind from the outside of the cell. When certain molecules bind, they favor a conformational transition, a structural transition of the GPCR from its inactive state to its active state. And then once the GPCR is in its active state, it can couple to various intracellular signaling proteins. And the classical example of such an intracellular signaling protein is a G protein. In fact, that's why they're called G-protein coupled receptors. So over the past few years, we have used molecular dynamics simulations basically to understand each of these three steps: the binding of ligands to GPCR, activation, and G-protein coupling. And that's what I'll talk about today. It's actually worth emphasizing that the structures shown here, these are actually all structures solved in Brian Kabilka's lab over the last few years. So up until 2007, there was a high-resolution structure for only a single GPCR, rhodopsin. And that's sort of an unusual one. It's activated by absorption of light rather than by binding of a diffusible ligand. And the techniques used to solve the crystal structure for rhodopsin didn't generalize to other GPCRs. Since 2007, there's been a real revolution in GPCR crystallography. Actually, not only crystallography, many other sort of experimental structural study techniques as well. So there's a great deal of information available experimentally that wasn't available just a few years ago. But those crystal structures give static snapshots of these GPCRs. And GPCRs are inherently dynamic. Their function depends on their ability to change structure. So we have focused on using molecular dynamics simulations to understand these processes and to understand the dynamics of GPCR function. I'm going to make one methodological point, although I said I wouldn't focus on methodology. So that point is just that each of the three steps I'll talk about here involves allostery. And one thing that's worth keeping in mind for allostery is that allostery is symmetric. What I mean by that is that if the binding of one ligand to a GPCR, to any receptor makes a second ligand bind more tightly, then binding of the second ligand will make the first ligand bind more tightly. Okay, a more rigorous statement of that is that the difference in binding energy of ligand A with or without ligand B is the same as the difference in the binding energy of ligand B with or without ligand A. Okay, this is not a new result, this is a very old result, but it's not always intuitive. Particularly, it's not always intuitive to reviewers. It's probably not always intuitive to audience members, so I thought it was worth pointing out explicitly early on. All right, so now we'll divide the rest of the talk, apart from the last little bit, by binding, and then activation, and then G-protein coupling. So let me start with binding, how ligands bind to GPCRs. So one of the things that's become possible with molecular dynamics in recent years, basically as simulations have gotten longer, is to actually simulate the full binding process of a ligand to its receptor. So what we do here is that we take a particular GPCR, an adrenaline receptor. The GPCR is embedded in a lipid bilayer with water on both sides, although the lipid bilayer and water are not shown in this movie. And then we take a drug molecule. This is a beta blocker, alpranolol. So it's a drug used to treat heart attack and hypertension. And we just place that drug molecule randomly in the water surrounding the receptor. And then we run a simulation. The simulation doesn't know anything. We don't tell the simulation anything about where that drug should bind. So initially, as you can see, it just sort of bounces around randomly. But then it works its way into the binding pocket. And once it gets there, it stays there for as long as we can simulate. Which makes sense because this simulation is a few microseconds long, and the experimental off rate of this ligand is minutes. So we don't expect to ever see it come off. Now, when we first did the simulation, there was no structure of this particular drug bound to this particular receptor. But subsequently, by coincidence, the structure came out from Ray Stevens' lab. And so we can compare where the drug ends up in the simulation to what's seen experimentally. And as you can see, it's a very close match. We can also use simulations to compute certain properties of binding that are easy to measure experimentally, like affinity and on rate. And those are also fairly good matches. The real power of these simulations, though, is in computing properties of the binding process that are difficult to get at experimentally. So in this initial study, we used simulations to study determinants of binding kinetics, what it is that determines the on rate and the off rate of the ligands. I'm not going to talk about that today. I want to move on to a more recent study, where we looked at what are called allosteric modulators of GPCRs. So allosteric modulators are basically ligands or drugs that bind anywhere but the classical binding pocket. The classical binding pocket is where you saw the beta blocker binding in the previous movie. It's right about here. So this is where the vast majority of native GPCR ligands, of your body's own GPCR ligands, and also GPCR-targeted drugs, that's where they bind. The classical binding pocket, it's also called the orthosteric site. Allosteric ligands, or allosteric modulators, are ligands that bind anywhere else. And there's a great deal of interest in them from a drug development perspective. There's a great deal of interest in them because they represent one very promising way to achieve selectivity between closely related receptor subtypes, which tend to be identical or almost identical in the orthosteric site, in the classical binding pocket, but more different everywhere else. That is, the residues in the binding pocket are more conserved, in the classical binding pocket are more conserved than the residues elsewhere in the receptor. Allosteric modulators also represent a way to basically, they offer the potential to modulate the body's own signaling patterns instead of overriding them with a drug that turns the receptor on or off all the time. In any case, there's been a lot of work on allosteric modulators, and a lot of allosteric modulators of GPCRs are known. But when we did this study, there were no crystal structures of any GPCR with a drug like allosteric modulator bound. And it was even, so it wasn't clear where they bound or how they bound, and it was even less clear how they exerted their effect on the receptor. And their main effect on the receptor is generally that they increase or decrease the affinity of the classical ligand, of the orthosteric ligands that bind in the classical binding pocket. So we set out to study that by simulation. So the first thing we did was to try to figure out where the allosteric modulators bind and what their binding pose is. And our approach to that was conceptually very straightforward. We took a GPCR. This is not the adrenaline receptor. It's a muscarinic acetylcholine receptor, simply because there have been a lot more allosteric modulators identified for this muscarinic acetylcholine receptor. There have been a lot of sort of a classical model system for experimental study of allosteric GPCR modulators. So we took this M2 muscarinic acetylcholine receptor, and then we took a known allosteric modulator. I'll refer to this ligand simply as C7, because it has sort of a long name. Actually, this C73PHDH is an abbreviation. It has a much longer name. So this ligand is known to bind allosterically. One can show pharmacologically that it binds allosterically just by showing that it binds non-competitively, does not compete with the classical ligands that bind orthosterically. But it wasn't clear exactly how it bound. So we simply, again, place this molecule in the water surrounding the receptor and then run a simulation. And again, it bounces around at random because we're not telling it where to bind. But eventually, it binds up here. So it's actually following the same pathway that the beta blocker followed to bind to the receptor. But it gets stuck partway through. Interestingly, the beta blocker also paused at right about this spot. And at the time, I wondered whether that could be an allosteric binding site. But it was only once this structure of a muscarinic acetylcholine receptor became available that we were able to do this study. So we've done multiple such simulations, and it binds repeatedly right about here. And you may have noticed in the movie that once it binds, this part in dark yellow is bound stably, whereas these arms in light yellow wave around like this. Okay? It's a hand-waving explanation. Okay, so there's still no crystal structure of this modulator bound to this receptor. So we can't compare to a crystal structure. But we can compare to mutagenesis data.
So wait a second, what does it do? It prevents the correct modulator from reaching its site, or blocks the passage?
So I'll come back to that in a moment. But in a nutshell, this particular modulator decreases the affinity of orthosteric ligands, but other modulators that we studied that bind in the exact same site actually increase the affinity of orthosteric ligands. So it can go either way. So it's not just a simple matter of blocking its axis.
So it's not a competitive inhibitor?
Right. So all of the allosteric modulators I'll talk about are not competitive inhibitors.
It blocks the entry.
It is possible that the orthosteric ligand can sneak by it, but my guess actually is that it can't. And then what happens is the orthosteric, so this modulator, its on rate and its off rate are fairly fast. So it binds and unbinds fairly frequently. So the orthosteric ligand can bind first, and then the modulator can come, and the modulator can, basically depending on the cooperativity, it can either increase or decrease the affinity. We can come back to this later. There are actually cases where the modulator decreases the off rate of the ligand, but also simultaneously decreases the affinity, which is a little bit counterintuitive.
And do you see a change in receptor structure?
Yes, I'll show that in a moment. Okay, but first let's just talk about validation of the bound pose. Like, is the bound pose correct? So we can compare to mutagenesis data. There have been a lot of mutagenesis experiments using this ligand and some very closely related ligands. And the general idea there is that people mutate many residues on the receptor, and they expect that residues that are in direct contact with a ligand when it's bound will tend to, when mutated, will tend to decrease the affinity of the bound ligand. So in fact, the 4 residues whose mutation substantially decreases the affinity of this ligand or very closely related ligand are the ones shown here in light blue. And they're all in contact with the bound pose that we see in simulation. And in fact, they're all in contact with this central part that bound stably while the arms were waving around. So that's encouraging. Now, we did simulations like this with several different allosteric modulators of this particular muscarinic acetylcholine receptor. And although they're chemically diverse, they have chemically diverse structures, they all bind in the same spot, and in fact, there's a common pattern to how they bind. So then, using our understanding of the bound poses, we went on to test these bound poses by making experimental predictions and trying them out. So what we did was to design several mutants that had not been made in the literature, that we predicted would either, in some cases increase and in other cases decrease the binding affinity of certain allosteric modulators. And we focused in particular on mutations that would increase binding affinity because it's harder to do that by luck. So the mutations that are shown here in yellow or red, so yellow here or red, or there are some that are a combination of the yellow ones and the red ones, those are predicted to increase binding affinity. The ones in blue are predicted to decrease binding affinity. Arthur Kostopoulos' lab at Monash University in Australia made these mutants and then measured the binding affinities, and in fact, in every case, the direction of change, whether it's an increase or decrease in affinity, is as predicted computationally. Okay, so bottom line is that we're reasonably confident about the bound poses of these modulators, which interestingly were different from guesses that were in the literature previously from earlier modeling studies. But what I haven't addressed yet, and actually what I find the most interesting part of this study, is what's the structural basis of allosteric modulation. That is, I mentioned earlier that the main effect that modulators exert is that they either increase or decrease the affinity of the orthosteric ligands. But how does that happen? So first, a bit of terminology. It turns out that, well, so C7, which is the ligand that I showed binding before, the modulator I showed binding before, that is what's called a negative allosteric modulator. That is, the presence of C7 decreases the affinity of the classical antagonist NMS. So I'm going to focus on allosteric modulation, increase or decrease. I'm going to focus on cooperativity with this particular ligand, which is probably the most widely used ligand in experiments on muscarinic acetylcholine receptors. It turns out that the cooperativity actually depends on which orthosteric ligand you're considering. Okay, so there's the most experimental data for this one. So C7 has negative cooperativity with NMS. It's thus referred to as a negative allosteric modulator. Some of the other modulators we looked at have positive cooperativity and are referred to as positive allosteric modulators. Now, as I mentioned earlier, the allostery is symmetric. So the fact that the presence of C7 reduces the binding affinity of NMS is equivalent to the fact that the presence of NMS will reduce the binding affinity of C7. And that latter statement is an easier one to examine in simulation because the allosteric ligands bind much more quickly than the orthosteric ligands. So what we did is to do binding simulations like the ones I showed you before for C7, but to do some with NMS present in the orthosteric site and some without NMS present. And in fact, we see the cooperativity that's measured experimentally. So what we see is that when there's no ligand in the orthosteric site, C7 binds reproducibly, goes to its canonical bound pose, and it stays there. What these bars represent, basically each of these bars represents one simulation, and the color coding denotes the extent to which the allosteric ligand, C7, is bound at each point in time. So basically the dark blue represents binding in the canonical pose. So you can see that each of these ends up with a ligand bound and it just stays bound. On the other hand, when NMS is present in the orthosteric site in the simulations, C7 sometimes still reaches its canonical bound pose, shown here in dark blue, but it doesn't stay there. It shifts to a different pose or comes off the receptor entirely. So we can see the negative cooperativity that's observed in an experiment. But now the question is, why is it happening? Yeah?
It seems that both the on and the off are affected.
Yes, both the on and the off are affected. Okay, but yeah, so let's get to what's going on. It turns out that there are at least two mechanisms. There are at least two mechanisms at play. The first one is conceptually very simple. The first one is just electrostatic repulsion between like charges. NMS is positively charged, has a positive charge of +1, so the orthosteric ligand is positively charged. And all the allosteric ligands we looked at, in fact, all the allosteric ligands that are typically studied for this receptor are also positively charged. They have positive charges ranging from +1 to +3. So you would expect that there would be some electrostatic repulsion between these ligands. Now, it's not clear that the magnitude of that electrostatic repulsion would be sufficient to explain any of the experimentally observed effects. And in fact, interestingly, as far as I can tell, there was no discussion of electrostatic repulsion as a possible mechanism of cooperativity in the literature on allosteric modulators of muscarinic receptors. But using this, computationally, once we know the bound pose and the conformation of the receptor, we can calculate what the magnitude of that electrostatic repulsion is. And for some of the ligands, in particular for C7, that electrostatic repulsion is basically sufficient to explain the experimentally observed cooperativity. But for other ligands, it's not. And the easiest way to see that is that basically like charges are always going to repel, not attract, so that you would always get negative cooperativity from that mechanism. But some of these modulators have positive cooperativity. In particular, alkyronium has essentially the same charges as C7, the same repulsion with NMS as C7, yet it's a positive modulator, has positive cooperativity instead of negative cooperativity. So there has to be something else going on there. So that something else appears to be a coupled conformational change between the orthosteric and allosteric sites. So this gets back to the question that was asked earlier about whether binding of these ligands affects the conformation of the receptor. So here's what we see. First of all, these are cross sections through the top of the receptor. And you see the orthosteric site here and the allosteric site here. So this is what the receptor looks like when NMS is bound and there's no allosteric modulator. If we take out the NMS, the orthosteric site becomes a little bit narrower That's not too surprising. But what is interesting is that the allosteric site becomes a lot narrower. It goes from a wide conformation to a narrow conformation. So now here are similar images, but where the allosteric modulator is bound. C7 bound here, and alkyronium, the positive modulator, bound here. Now you'll notice that alkyronium is a sort of wide, bulky ligand, and it actually requires the allosteric site to be in a wide open conformation in order for it to bind. Now if NMS is present, then it basically pre-opens the allosteric site, and that makes it easier for alkyronium to bind. And if NMS is absent, then in the absence of alkyronium, the allosteric site will be narrow. Alkyronium basically has to force it open, which takes energy. So the presence of NMS makes it easier for alkyronium to bind, and likewise the presence of alkyronium will make it easier for NMS to bind. C7, on the other hand, is a narrow ligand, and it's perfectly happy binding in either the closed or the open conformation of the allosteric site. We wanted to test that mechanism experimentally. So what we set out to do was to start with C7, which is the allosteric modulator that I showed binding in the movie, and to try to modify its chemical structure in a way that would essentially try to modify its chemical structure in a way that would shift its binding cooperativity in the positive direction. So the idea is that we designed computationally a variant of this molecule that requires a wider conformation of the allosteric site in order to bind. It's not as wide as alkyronium, and we don't necessarily expect the cooperativity to be positive. We just expect it to be less negative than C7. And in fact, that's what's seen experimentally. So Jonathan Bell's lab, also at Monash, synthesized this ligand. Arthur Christopoulos' lab measured the cooperativity with NMS. And in fact, the cooperativity is significantly less negative or more positive than the cooperativity of the ligand we started with. This is actually the first example I know of basically of structure-based design with the goal of shifting the cooperativity of an allosteric modulator for GPCR. So that's all I'm going to say about ligand binding, allosteric and orthosteric ligands. I want to move on and talk a little bit about receptor activation, the process by which the receptor goes from its inactive state to its active state. So the easiest way to approach that conceptually would be to just do simulations where you take an inactive conformation, put an activating ligand, an agonist, in the binding pocket, and then just run a long simulation. And you might expect the receptor to activate. So we tried that, and it didn't work. But later, after we tried that, Brian Kubilica's lab solved two structures, basically the first two structures of a GPCR with an agonist bound. And the interesting thing about them was that one of those structures looked active, the other looked inactive. Both of the structures had an agonist, a strong agonist, clearly bound in the orthosteric binding pocket. There were various differences between the structures. One difference that we focused in on was the fact that the one that looked active, the one shown here in red, has this outward shift of helix 6, which had on the basis of various biophysical experiments, had been associated with activation of GPCRs. So that structure looks active, and that structure is stabilized for crystallization in part by the presence of a G protein mimetic that binds on the intracellular side. The other structure, shown here in gray, it essentially perfectly matches the inactive structure. Okay, and that one doesn't have the intracellular binding partner, the G protein mimetic. And in fact, it turns out in simulation, if we do simulations where we start with the structure that looks active, that is now on the basis of a lot of evidence believed to be the active structure, so the red structure, we remove the nanobody, we remove the G protein mimetic that was bound on the intracellular side. Then what happens is that in simulation, we see spontaneous deactivation, the spontaneous transition from an active state to an inactive state, even though the agonist is present in the orthosteric binding site. So let me show you that. We start the simulation in the red state, and basically what happens is it spontaneously discovers the inactive state of the receptor, which is shown in gray. We're not telling the simulation what the inactive state is, it just gets there spontaneously. Now, our initial motivation for doing this study actually was that we were working with Brian Kabilka, and he was trying to understand why one of his structures looked inactive. And one hypothesis that we wanted to test was the notion that you actually needed the intracellular binding partner to make the active state the dominant state, as opposed to a minority state that you won't see crystallographically. So that really appears to be the case. But now, using simulations like this, we can actually study the activation process. Because what we're simulating here is essentially activation in reverse. We're simulating the transition of an agonist-bound receptor from its active state to its inactive state. And so by doing such simulations repeatedly and looking at the process of the transition, we can characterize the activation process. So in a nutshell, here's what we find. We find that, so essentially we're trying to understand how the signal is transmitted from the classical binding pocket, the orthosteric site, to the G protein binding site on the intracellular side of the receptor. This is also an allosteric mechanism. Allosteric in a different sense from the allosteric modulators I talked about before, but there's communication at a distance between the binding pocket and the G protein binding site. So what we see in simulation is that there's coupling between three regions of the receptor. The binding pocket, an intermediate region that we call the connector region, and the G protein binding site. And each of those three regions transitions in simulation between 2 or 3 well-defined conformations. Now, interestingly, the coupling between them is quite loose. They don't move in lockstep, like interlocked gears. Instead, each of them, so basically we see many different combinations of conformations of these different sites, almost all the possible combinations of conformations. But if one of the sites is in a particular conformation, it sort of shifts the probabilities that the neighboring sites are in a particular conformation, so you still are able to transmit a signal through the receptor. One thing we've looked at more recently, actually these are results that just came out over the past year, is an interesting question of how basically the similarities and differences between activation mechanisms of different GPCRs. So the thing that's interesting here is that if you look at all the GPCRs for which inactive states and active states have been determined, the conformational transition between the inactive state and the active state is strikingly similar. It's generally similar not just on the intracellular side, but also in the binding pocket. Yet the ligands that bind to these GPCRs and activate them are dramatically different. So the family of GPCRs, different GPCRs in the family, sense the presence of vastly different ligands. They've evolved to sense the presence of vastly different ligands. So the question is, these different ligands that cause a similar conformational change on activation, are they acting in similar ways or in different ways? And the answer, at least for the ones we've studied, appears to be that they're acting in very different ways. So in particular, we've looked recently at 2 different receptors. So we looked earlier at the beta-2 receptor, an adrenergic receptor. That's the one I showed you a moment ago in the movie. But more recently, we've looked at the mu-opioid receptor, together with Brian Kabilka, and a chemokine receptor, US28, with Chris Garcia. So Chris Garcia's lab is next door to Brian Kabilka's, and after I arrived at Stanford, I discovered that he had basically also started to crystallize GPCRs. Sort of a competition between these 2 labs that are next door to one another. So we worked with both of them on 2 different receptors, and what we found in each case is that the activation mechanism is, or that what the ligand does to trigger activation is different. So for example, in beta-2, the ligand pulls on helix 5. In the mu-opioid receptor, it basically pushes on helix 3. So the ligands do different things, and yet they lead to similar conformational changes. Okay, and I'm going to skip the slides kind of showing all the details of that so I can get on to the next topic. So the next topic, so the last major part here, is using simulations to understand the mechanism of G protein coupling. So basically, how does the receptor cause activation of the G protein? So first, let me sort of give you what's been known for decades. So the question is, how does a GPCR trigger G protein signaling? The basic answer to that, and this is what you'll get from a biology textbook, and it's correct, is that the GPCR triggers G protein signaling by causing GDP to dissociate from the G protein. So the G protein, in its resting state, has GDP bound. The GDP can come off, but very, very, very slowly. once a GPCR binds, once an activated GPCR binds, the GDP comes off many orders of magnitude more quickly. And then once the GDP comes off, a whole bunch of other things happen. So once the GDP comes off, GTP binds, then the G alpha and G beta gamma subunits separate, and they initiate a whole bunch of downstream signaling. But all we really need to worry about for now is what the GPCR does. And what the GPCR does is simply to dramatically accelerate release of the GDP. The question is how that happens. Okay, what was not clear is how that happens. Now, one major step toward answering that question was this paper that came out at the end of 2011. This is the first and actually still the only structure of a GPCR bound to a G protein. It's probably a big part of the reason that Brian Kabilka won the Nobel Prize the following year. So this structure is useful, but it still doesn't tell us what's happening. In fact, it raises some major questions. So all the structures of G proteins, except for that complex structure that was on the cover of Nature, all the other structures look like this. So they all have a nucleotide bound, GDP or another nucleotide. That nucleotide is bound right here, and it's sandwiched tightly between these two domains of the alpha subunit, the Ras domain and the helical domain. Now, Brian's structure of the complex is different, and it's actually different in two ways, three ways. So the first is that there's a GPCR bound. The second is that there's no nucleotide bound. The nucleotide has already escaped. And the third, as you can see, is that there's this dramatic conformational change, this dramatic separation of the Ras domain and the helical domain, which go from being like this to being like this. It's a motion of about 45 angstroms. Now, that raises several questions. Actually, it raises one big question, which is, what is the receptor actually doing to cause the GDP to escape? Several possibilities were suggested in the literature. The first is that the receptor causes that opening motion, and that opening motion allows the GDP to get out. Right, so instead of being tightly sandwiched, it's sort of this more open conformation, then the GDP can escape. Another possibility is that the receptor forces out the GDP. First forces out the GDP between the domains, just sort of through a back door, and then once the GDP has escaped, then the absence of the GDP causes that dramatic separation. Our simulations actually suggest that the answer is not quite either of these. So there's a third possibility. Okay, so let me explain that. Here's the first major observation from simulations. The first major observation is that when we take one of these one of these structures, one of the older structures with no receptor, no GPCR, and with a nucleotide bound, and when we simulate it, we actually see spontaneous domain separation. So recall that in all of these structures with a nucleotide present and the GPCR absent, the domains are these two domains, the Ras domain and the helical domain, are pressing against one another. When we simulate, we see that there's actually quite a bit of mobility. They fluctuate between a closed conformation and a more open conformation. They never go to a conformation that's as open as what's seen with the receptor bound and the nucleotide absent. They just sort of fluctuate like this. But they visit conformations that are plenty open enough for the GDP to have a clear exit pathway, as you can see here. Yet in these simulations, the GDP does not escape. It remains bound. In fact, if we delete the helical domain entirely and run a simulation, the GDP still remains bound. So the GDP is bound pretty tightly to this Ras domain, even in the absence of the helical domain. Now,
When it's moving to the Ras domain of the white protein important, does this happen?
not really. The white is the beta gamma subunit. It's included in all our simulations. It does move quite a bit, which is sort of interesting. That's interesting in its own right, but it's not. I don't believe it's relevant to the mechanism that we're going after here.
But does it allow you to explain beta gamma release on ATGT?
No. So we have right. So to study beta gamma release, you would need to, as you said, study the GTP binding process. We have done a few simulations with GTP bound, but there we started with the GTP bound conformation, so the beta gamma were already released. So basically, we haven't studied that process. It would be interesting, though. Okay, so now, what if we reduce the affinity of the nucleotide to the Ras domain? One way to do that is to look at GMP instead of GDP. So GMP is basically GDP with one phosphate group removed, and it forms fewer contacts with the Ras domain. So when we simulate with that nucleotide, it in fact escapes. It escapes fairly quickly, but it always escapes after the opening takes place. So it appears that the opening, the spontaneous opening, is necessary for release of the nucleotide. And in fact, if we do simulations just like this one, but where we artificially restrain these two domains so that they can't separate very much, then the GMP does not escape. So basically, what we can conclude, actually, let me show you one more thing. So what if we just delete the nucleotide entirely and simulate? If we remove the nucleotide entirely, just take it out before we run the simulation, then we see more dramatic domain separation. In the absence of the nucleotide, the G protein goes to a conformation that's very similar to what's seen crystallographically with the receptor bound. Right? So in the receptor bound structure, there's also no nucleotide, and there's this dramatic separation. So the simulations so far basically suggest the following. They suggest that you need, well, there's this spontaneous domain separation. That spontaneous domain separation is necessary for nucleotide release. If you prevent it, the nucleotide is not released, but it's not sufficient for nucleotide release. For the nucleotide to escape quickly, you need to somehow weaken the affinity of the nucleotide for the Ras domain. Okay, so that suggests that what the receptor is doing is somehow causing a structural rearrangement of the Ras domain that weakens its affinity for the nucleotide. When the nucleotide's affinity is reduced, then once the spontaneous domain opening happens, the nucleotide can get out. And then once the nucleotide is out, once the nucleotide is absent, then the domains open further to the structure that's seen crystallographically with the GPCR bound. But there's still one major question, which is what's the GPCR actually doing to the Ras domain to weaken its affinity for the nucleotide? So how can we study that? We could study that by just trying to simulate the whole process of receptor G protein association and then looking at what happens to the Ras domain when it associates with the receptor. But that's a very slow process. We're not able to capture that in simulation. So instead, we took sort of a more indirect approach. The approach we took is basically to compare simulations of the G protein with and without GDP bound. Now here's the rationale, and it actually goes back to this symmetry of allosteric slide that I showed at the very beginning. The rationale is that the G protein, basically if a particular G protein conformation reduces the affinity of GDP, then we would expect the removal of GDP to favor that conformation. So we expect that we would see that conformation more frequently in the absence of GDP than in the presence of GDP. And in fact, we do see differences.
Aren't there crystal structures that show that?
No, because the only crystal structure, the only crystal structure of a G protein with no nucleotide present is the GPCR bound one. And that has the GPCR bound, so it's more has happened than just removing the nucleotide.
What's all the crystal structures of G protein with?
It's actually really hard to work with it biochemically. It turns out the G proteins in the absence of GDP are over periods of minutes or hours, they're not very stable.
Well, that in itself
So they yeah.
And there is no mutants or mutants of GPCR which have very similar combination without nucleotide?
Mutants of GPCR or of G protein?
GPCR.
But I mean, so the issue is that experimentally it is just difficult to study the G protein without the nucleotide. There's no crystal structure. There's one paper in the literature that claims to have done NMR studies on it. I spent a lot of time studying that paper and talking with other people who have done NMR on G proteins. Basically, most people just don't believe that paper. In fact, I'll talk a little bit later about the experimental validation we did. Our initial ideas for experimental validation involved studies of a nucleotide-free G protein, and basically they all failed after more than a year of effort. the experimental lab, so Roger Sunahara's lab that we were collaborating with, sort of a G protein expert, he just couldn't get it to be stable enough to do any of the experiments we wanted to do. Okay. So let's go back to this question of differences we see in the G protein with and without the nucleotide. The primary difference we see is in this helix. This helix is called the alpha-5 helix. And in fact, it turns out to be the helix that contacts the receptor most closely in the receptor-bound state. I'll come back to that. First, let's just look at what happens to the alpha-5 helix when we remove the nucleotide. So here we're zooming in. This is the alpha-5 helix. This is the nucleotide. So now we remove the nucleotide, and then we run a simulation. So in a number of the simulations of this G protein with the nucleotide removed, we see a notable conformational change. So you'll see this helix shift. It will shift upwards. Okay, so it's shifted, and at the same time it's sort of rotated. Now, this is the same helix, and this image is showing how it docks to the GPCR. Okay, and the important thing is that this helix forms direct contacts with this groove of the GPCR, and it's able to form those contacts because it's in this shifted conformation, this upwards shifted and rotated conformation. So now let me show you, I'm going to show you the same movie that I showed before, the same conformational change. But now I'm superimposing on this structure the green structure, which is what the helix looks like when it's bound to the receptor. But we're doing the simulations in the absence of the receptor, just with the GDP removed. So we start from the red conformation, and basically it will go spontaneously to the green conformation, the receptor coupling conformation. Okay, so you see it's shifted to a conformation that would allow it to couple to the receptor. So what have we learned here? It's basically that this conformational change, which was already known to favor receptor binding, and thus to be favored by the presence of the receptor, also reduces affinity for GDP.
And if you run that with GDP present, doesn't happen.
Yeah, so when we run with GDP present, it happens at least 1,000 times less frequently. So there are a few frames in those simulations that are similar to this, but they're much, much, much less common. Okay, so one way to further test this, one way to test this computationally is to force this conformational change in the helix, in the alpha-5 helix, in simulation. And we would expect that when we force that alpha-5 conformational change, that will cause the GDP to come out faster. Now, it does, here we're doing it, but there's one important caveat. The caveat is that when we tried this in plain unbiased simulations, basically, the escape of GDP is fairly slow on simulation timescale. So when we just run a simulation, we don't see it escape. So we did a trick here to help it escape faster, and that's to use something called temperature-accelerated molecular dynamics, a technique from Eric van den Eynden, that basically involves heating just the ligand, heating just the GDP. Okay, in a sort of mathematically sophisticated way. So you have to ask, well, maybe it was just that heating of the GDP that made it come out. What if we just heated the GDP and didn't twist the alpha-5 helix? So the answer is, when we just heat the GDP and don't twist the alpha-5 helix, we don't see it come out. It just stays bound. So to summarize, here's the mechanism that we end up proposing. The mechanism for what the GPCR does to the G protein. Basically, when the G protein is on its own with a nucleotide bound, with GDP bound, and with no GPCR, it's an equilibrium between two states, a closed state and an open state. Here the domains are together, here the domains are separated. When the receptor binds, the receptor causes a local structural change in the RAS domain. You still have this equilibrium, the same equilibrium as before, but now the GDP's affinity to the RAS domain is reduced, so that when the G protein is in its open conformation, the GDP can get out. And then once the GDP has come out, that shifts this equilibrium toward the open state. And so that's what's seen crystallographically. So now finally, we wanted to test this experimentally. I'll show you that in 2 slides, but it took more than a year. So we ended up doing 2 successful experiments to test, as well as the other experiments that I mentioned previously, where we just never got any results. So the first experiment is just asking the question of, is there in fact an equilibrium between a closed and an open state with GDP bound? There was a little bit of evidence suggesting that there might be from previous EPR experiments. So we basically did additional EPR, in particular DEAR, double electron-electron resonance experiments that provided more evidence for this. So the way these experiments work is that labels are inserted in the Ras domain and the helical domain, and the experiment measures the distribution of distances between those labels. Essentially, like a histogram of the distances, averaged over a large population. And what you see in those histograms are 2 peaks. A large peak that corresponds to the closed conformation, and a small peak that corresponds to the open conformation. And we actually see it in 2 different G proteins that we looked at. So that's nice, but it still leaves the question of, is this functionally important? Can we show experimentally that that spontaneous opening is functionally important? Because you can imagine a situation where the open conformation that's seen experimentally is just some like minority, some fraction of the sample that's in some degraded conformation, and that's just not functional. So the experiment that shows that it is functionally important is a second experiment where we're looking at the off rate of GDP. We're actually looking at nucleotide exchange rate, but under conditions where it's limited by the off rate of GDP. And we look at 2 constructs. One is just the wild type G protein. The other is an engineered G protein where this tether has been added to basically tie the helical domain to the beta gamma subunit in a way that makes it harder for the helical domain to open. The helical domain will still open. This is not a covalent link. It's just held there by a weak protein-protein interaction. But basically, we'd expect the helical domain to open less. We'd expect the G protein to be in a closed conformation more of the time. And in fact, what we see there is that the off rate decreases substantially. The off rate of GDP decreases substantially. So this supports the notion that in order for GDP to come off, in the absence of a receptor, it has to wait for a spontaneous opening event. Okay. So I'd like to show 3 more slides. This last little bit is methodology focused. It's actually motivated. I decided to show this because of something Michael mentioned in his talk, where he mentioned that analysis of MD simulations is still a difficult problem and still not really solved. And actually, I completely agree with that, but I want to describe one piece of work we've done recently, one technique we developed for analysis of simulations. Doesn't solve all the problems, but I think it's useful. So the question we were looking at is how to automatically detect important conformational changes. Important conformational changes in a simulation. For example, the detailed conformational changes involved in G protein activation. So one way you might approach this is using change point detection. There's a large statistics literature going back decades on change point detection, which is basically you're given a time series measuring something, and you want to find a point in time where the statistics of that time series change. Maybe it's mean changes, maybe it's variance changes, maybe something else changes. People have developed a lot of ways to do that. So you can imagine applying change point detection to this problem. What makes it hard is that you don't have one time series, you have many, many time series. And the changes often affect only a few of them. So you might look, for example, at distances between pairs of atoms in the protein. But then there are a lot of distances to look at. And a particular conformational change, a large conformational change might affect many of them, but some of the important conformational changes involve only 1 or 2 residues. And those are going to affect only a few of these time series. So there's sort of a needle in a haystack problem. And traditional techniques for change point detection don't work well. Actually, before I tell you what we did, let me just show you this movie, which illustrates the results of the method. So this is a simulation of a GPCR going from its active state to its inactive state. And the movie highlights the changes that are detected by our method as being most important. And you can see that some of them, so each of the changes is a blue to red shift. And the first couple are very subtle shifts. They involve only a couple residues. Okay, the distinctive thing about them is that they're long-lived. They happen rarely. We're looking specifically for changes that happen rarely, not for residues that are just waving back and forth the whole simulation. So how does our method work?
Well, in doing that,
Yeah.
you actually focus on residues, you just focus on everything.
We focus on everything. We just look at all the time series. And what we try to do is find points in time where there are changes and which time series change at those points in time. So the results of the method are change times and which time series change at those times. Once you know which time series change, it's relatively straightforward to map them to individual residues. But that's a post-processing step.
How do you know if it's important or not important?
Let me come back to that. Let me just explain what the method is doing. But I'll give the sort of mathematical definition in a moment, but our conceptual definition is that we're basically trying to represent the trajectory by saying that the statistics are constant, except for a few points in time where there are changes. And then we try to look for what satisfies that the best. So essentially we're looking for rare changes. The intuition is that the important conformational changes are rare changes. Those won't always correspond to biologically important changes, but the intuition is that they often will. Okay, so here's the basic idea of the approach. So we're going to approach this as a statistical change point detection problem, but instead of trying to determine just change times, we're going to look simultaneously for change times and which time series change at those times. We end up formulating this as a giant optimization problem. It's an optimization problem that on the face of it looks like we wouldn't be able to solve it. It turns out that there are some recently developed algorithms that we can apply to this problem to make its solution practical. So here's a more mathematical description. We start off with a set of time series. In reality, there are usually thousands of them or tens of thousands, but in this example, there are just 4. And basically what we're going to do is consider different change points. And here we have change points for each of the time series, some simultaneous, some not. And for each set of candidate change points, we have a measure of quality. And then we're going to maximize that measure of quality. We're going to find the change points that maximize the measure of quality. So the measure of quality has 2 parts. The first part, we basically say, okay, given the set of candidate change points, let's fit a simple statistical model to each segment of each of for each of these observables, for each of these distances, we will fit a statistical model to each segment. And a segment is just the time points between hypothesized change points. We fit that with a simple statistical model, and then we look at the likelihood of the data under that model, and we want that likelihood to be as high as possible. So that's one of the terms that will go into the function we want to maximize. The other term is a penalty function, and the reason we have this penalty function, the motivation for this penalty function, is that we want to explain the data with as few change points as possible. Right? Like we could get very high likelihoods if we say that there are change points, there are a million change points, there are change points all the time. But that's not a very useful model. We want a parsimonious description of the data. So we want the number of change points to be as small as possible. So we have a penalty function that penalizes us more when we have more change points. But we also know that when conformational changes take place, they tend to affect more than one time series simultaneously. So we're going to penalize additional change points less if they happen simultaneously than if they don't happen simultaneously. So then we combine these 2 parts into this objective function that we maximize. And then actually maximizing it is not easy. It turns out that there's a very clever algorithm that was introduced recently by another group in the context of single time series change point detection. It's actually guaranteed to find the best solution in that case. We applied it in an iterative fashion in a way that is not guaranteed to find the global optimum, but in practice it seems to work very well, and it's relatively fast.
Aren't there a huge number of change points?
So actually, there's a sensitivity parameter here. There's this lambda parameter is a sensitivity parameter. Basically tells you what weight you place on the penalty function. The more you weight the penalty function, the fewer change points you will find. So what we typically do in practice is we run the algorithm with different values of the penalty function. And the change points that are detected at the largest values of the penalty function, that we consider those the most important. Alternatively, you can think of those as the slowest timescale changes. When you go to smaller values of the penalty function, you also detect the faster timescale changes. So if you go to very small values of the penalty function, you detect a huge number of changes. And yeah, I mean, that makes sense intuitively because there are lots and lots of changes. But you want a way to pick out the most important ones. So you could use either all the coordinates or pairwise distances. If you use the coordinates, you have this alignment problem, right? The whole protein will typically be tumbling, like rotating and translating in space. So you have to align it, and then you have a question of what to align it on, like which atoms to use for the alignment, which is actually kind of tricky. If you use pairwise distances, you don't have that problem. On the other hand, you have more pairwise distances, so there's a trade-off. In practice, we have done this primarily with pairwise distances. We did try doing it with atom positions. Also, it works reasonably well. You could also do it with dihedrals, with dihedral angles. That's actually quite attractive. The one tricky bit of doing it with dihedral angles is now you have this rotational effect that they're circular coordinates. You could take a sine and a cosine.
I think pairwise distances are the right thing, because interactions depend on pairwise distances.
Yeah,
So
that's
I
true.
think it's
That's true.
Exactly right.
Yeah.
And don't worry.
Okay.
What do you mean by simultaneous change? Does it mean that you see the changing point at a similar time across different time trajectory?
Across different time series.
Yeah, so that is defined simultaneous.
Yeah. I mean, so the basic idea, like the simplest thing you could do, you could just take each of your pairwise distances and put them through a traditional statistical change point detection algorithm. But it's not going to work that well because there's limited information in each individual time series. And the conformational changes that you care about, even if they involve just a single residue, they're going to affect more than one time series. So really, you want to focus on changes that affect more than one time series. So we call that simultaneous change point detection. Yeah, and then we came up with a name so that we could call it the simple method. All right.
So one last question. Do you reduce it according to the distances, or is it every atom pair?
It depends on analysis. So if we're looking at a protein folding trajectory where things are sort of changing all over the place, then we use all of them. If we're looking at a simulation of a folded protein where we know that we're going to be looking for more subtle changes, then we usually sort of pre-filter. We just essentially find atoms that are near one another at any point during the simulation and only use those distances, because basically we know that what's going to be important is basically changes that are going from contact to no contact. You can also apply a nonlinearity to the distances to really kind of highlight changes in the regime between contact and no contact, like around 4 angstroms. Yeah.
So the underlying assumption all the time is that the important changes are the rare ones.
Yes.
And so
And also the changes happen fairly quickly. And actually, I see those as sort of the big limitations, especially that last one about changes happening fairly quickly, as the big limitations of this method. So there's kind of an underlying challenge here, which is just how do you define mathematically what is important in an MD simulation? So we started with one definition, and we solved that problem, and it's useful, but there are things that are interesting in MD simulations that don't fit that definition.
expect from a biological, functional perspective for the functionally important changes not to be the rare ones?
Oh, no, I would expect functionally important changes to be fairly rare. At least rare in simulation timescales, right? So so a simulation, like a long simulation is a few microseconds, right? Or I guess for the longest Anton simulations, a millisecond or 2 milliseconds. Those are, well, so by day-to-day standards, those are very short timescales. But basically, if you have an amino acid that's sort of waving back and forth every nanosecond, that's unlikely to be important. It's unlikely that I mean, you typically are looking for something that's going to trigger some reaction. It's not going to happen every nanosecond. Yeah.
All reactions involve going over barriers, so by definition they're going to be rare and happen quickly.
Actually, yeah, that's a very good.
And I think that there's no way you can get over a barrier slowly because you've got to jump. That's why most conformational changes are quick. You wait a long time, so they're rare and.
You could have a diffusive barrier, which, okay, but I agree with you. I was sort of trying to. I don't want to make it sound like the MD analysis problem is solved. So I was sort of highlighting some of the limitations here. But yes, I mean the intuition for this, and actually
I'm really excited about this. I never heard about this until 5 minutes ago.
Okay, but actually, Michael, the original motivation for this came out of discussions with you in 2009. Or 2008, maybe? A long time ago. And then we sort of used other approaches. We tried other approaches for about 3 years before trying this approach. So it's sort of a long path.
The penalty is not a function of the temperature. It has to be.
Okay, yeah, that's a good question. The simulations are, actually most of the simulations are body temperature, but body temperature, room temperature are fairly close. Is the penalty a function of the temperature? You could.
Physically, it has to be.
Well, hang on. Okay, so you can apply this method without knowing what temperature the simulations will run at. Right? Like to apply the method, you don't need to know anything about the force field or how. You don't need to know anything about how the simulation was run. You're just given the results, and you want to detect the changes. Okay. But as I said, you have this sensitivity parameter. I believe one could relate the sensitivity. Thinking back to the physics of the simulation, one could relate the sensitivity parameter to temperature. You can relate it to barrier height. And like if you think about each of the conformational transitions as a barrier jumping event, then the time scale will depend both on the height of the barrier and the temperature. And maybe on other things also. So there is an intuitive connection to temperature, but you don't need to worry about temperature when applying the method. Right? It's just a mathematical method that you apply to the results of the simulation and that detects changes. Okay, with that, let me wrap up because I'm out of time. So a very, very quick summary is that basically used all atom molecular dynamics simulations to look at the key steps in GPCR signaling, so ligand binding, activation, and then activation of the GPCR, and then how the GPCR causes the G protein to activate. And I've also talked a little bit about a method for detecting essentially changes in multivariate data. It's not even really specific to MD simulation. And finally, let me just thank a lot of colleagues and collaborators. A lot of the work I described was initiated at DE Shaw Research, and a lot of it was finished at Stanford, some of it was done at Stanford, some of it was initiated at Stanford. It also involved a lot of collaborations with experimentalists at Monash, at the University of Michigan, at UCLA, and at Stanford. Okay, great, thanks. I've never actually heard that song. It's just what comes up nowadays when you Google hello to find out whether you have an internet connection.
Thank you. Maybe one last pressing question.
I'm just wondering, because there's some evidence that the composition of the lipid membrane and the membrane itself can affect the function of the receptor. How difficult would it be to actually model the membrane as well in the simulations explicitly?
The membranes are modeled explicitly in all our simulations. I didn't show them in the movies. I also didn't show the waters, but there's an explicit membrane in all the simulations. And we have tried simulations with different membranes. We've tried simulations with different membranes, but we haven't focused on I have not focused on the effect of the membrane. There have been studies of that. You're right that experimentally, the composition of the membrane does have an effect. The effect is relatively subtle. So if you look at, for example, the rate of activation changes by a factor of 2 or 3, sometimes a little bit more, when you change composition of the membrane. But what we were looking at here was primarily sort of qualitatively, what's the mechanism? And I would not guess that the mechanism would change. And in fact, when we've done simulations with different bilayer compositions, we have not seen changes in the mechanism.
And when you do the simulation, do you always only have one receptor, or do you also take into account the oligomerization state or something?
In the simulations I showed you, there was one receptor. I have done a few simulations of multiple receptors. I've done a few simulations of multiple receptors. I have not focused on that and don't have any deep results along those lines. There are other groups that have done quite a bit in terms of computational study of receptor dimerization or oligomerization.
What force field do you use?
Charm. So that
And are the interactions of small molecules with the targets primarily electrostatic?
no, electrostatic and van der Waals. Yeah, they're such a good question. I've never tried to do sort of detailed breakdown, but the electrostatic interactions and van der Waals interactions are both important. It's not easy to separate them out.
Constantly.
Okay.
This is partial charges. It's not just electrostatics. All the hydrogen bonding is done by electrostatics. So basically, there's a lot of dipoles that are created by electrostatics. And there's not just net charge electrostatics.
So why does it work so well?
Very careful.
Yeah, it's very carefully tuned. So
Specific to the problem.
Conformational check.
yeah. I mean, I've used different force fields in different studies. The force fields used in these studies were charm, basically because it has the best bilayer models. At least historically, it's had the best bilayer lipid models. Okay, but there are many versions of charm, and so we've done some other studies sort of looking at force field quality, and it appears that the most, basically charm and also amber have improved a lot over the years. So if you use the most recent versions, they're significantly better than the older versions.
Ron,
One last question.
I'm wondering if you did any principal component analysis to see if the conformational change is intrinsic to the system. Because that would be kind of interesting.
Normal mode analysis.
Yeah.
I've done a little bit of PCA on the molecular dynamics results. I have not done normal mode analysis. Others have done normal mode analysis. Actually, Abraham, Dr. Abraham Sampson, who has done a bunch recently. Yvette Bahar did some years ago on rhodopsin. They do see. So my impression from reading those papers is that there definitely is a connection. At least some of the global conformational changes observed in activation, you see them in normal mode analysis. Everyone said me,
Well,
okay, so I still haven't gone through all the details of your recent manuscript, but at least in Yvette Bahar's older published work, she sees some of the sort of global changes. She doesn't see many of the detailed changes that I believe are also
no,
important.
but you said that the motion is intrinsic even when you don't have the receptor. So you should be able to see that from principal component analysis as a major. One of the top modes would be, say, that helix moving upwards and things that you see.
Well, let's talk about it offline. I mean, I agree that it's very plausible you would see it. I am not convinced you're guaranteed to see it. Yeah.
Just as a tiny point, I just got an email from Ja, our new shared postdoc.
Uh-huh.
He wants to start in normal mode.
Okay, great.
Thank you.